dance by the water neath the Mexican sky. Drink some margaritas by a string of blue lights. Listen to the mariachi play at midnight. Are you with me? Are you with me? Water neath the Mexican sky. Drink some margaritas by a string of blue lights.
Hello, and a terrific morning. It's really my pleasure to introduce Hervé. It is like a dream come true. I'm like a fan girl or something, but to me this is like having the Rolling Stones equivalent in science next to me. To introduce him, instead of saying where he studied, where he did his postdoc and all that, I would like to emphasize what he did for science, and it is also in a very nice way trying to explain something he could not understand. It's not that it was written on some project or something like that. It was not an objective of a project. It was just a curiosity. He could not understand why, while putting transgenes in plants, sometimes they would stop being expressed, and this silencing was spreading through the plant gradually. And that reminded him of how sometimes plants can recover from virus infections, that it is also like something that spreads gradually through the plant. So he thought, perhaps it's the same phenomenon. And then he pursued trying to understand how this works, and he could corroborate his idea that it was indeed the same phenomenon. Is very important, now we know all, for plants to defend against viruses and transposons and all these things, and that is in big part thanks to him. And yeah, he's got a lot of awards and a lifetime award achievement from INRAE, and now we have him here. Thank you for coming.
Thank you very much, Laura, for this kind introduction, and thank you for inviting me to tell you about the journey of my research. Thank you. Yeah, so what I'm going to tell you is really a journey, because as I said, Laura, this didn't start as a project that was written to try to understand something. It was just a series of discovery, and unexpected discovery. And so I started 40 years ago, exactly. I was a undergrad student, and I did the first transgenic plant in my institute. We were back in 1985, so it was just the starting point of making transgenic plant. And it happened that the first transgenic plant that I made were silenced. So of course, my supervisors told me, you did something wrong, of course. You have to redo it. And I did the experiment so and so. And each time I was getting this very surprising result of plants that were carrying the transgene but not expressing it. So for a long time, the time of my PhD, in fact, people were thinking I was doing things wrong until this first publication came. That was quite reassuring for me, because these two publications that came back to back in 1990 were showing exactly the phenomenon that I was observing on my side, alone, in France. And it was showing that when you try to overexpress an endogenous copy, here it was in Petunia the chalcone synthase, you had extra copies driven by the strong constitutive promoter 35 phase deriving from a virus, which is quite important. You were getting some time overexpression, but many times you were obtaining the opposite effect. This was not really called silencing at that time, it was called co-suppression, because the author thought that it would be nice to tell this phenomena co-suppression, because it's the suppression, the coordinated suppression of the endogenous gene and the homologous transgenes. So at that time I was quite reassured, thinking that what I was observing was something that actually happened in other cases than the one I was studying. And I was very lucky, in fact, because I had a system that I'm going to present now that was more appropriate to study this phenomenon, because as you can see here in Petunia, you could only see this phenomenon appearing at the end of the life of the plant, when it starts to flower, and so it's very late in the development. The system I was using was based on an enzyme called nitrate reductase, which is the first enzyme of the assimilation of nitrate. It reduces nitrate into nitrite, which subsequently is reduced into ammonium. So we were trying to reduce the input of nitrate in fields actually trying to reduce the input of nitrate to get the same yield of plants. So for that, we thought that if we increase the expression of nitrate reductase, we could get plants that would grow as much as a wild-type plant, but with less nitrate in the soil. And we got this phenomena that here is visualized by this yellowing of the plant, which in fact result from the silencing of the nitrate reductase. So we made many transgenic lines that were carrying extra ectopic copies of nitrate reductase, again under the control of the 35S promoter. And at that time, it was quite easy in France to make field trial. We were at the beginning of the end of the 80s, the beginning of the 90s. And we could do very large-scale analysis of a large number of transgenic lines. And I'm going to show you just a few representative ones. And what is very important is that this phenomena takes place at each generation. And it does not affect, it's not an all or nothing phenomena. It can affect either the entire population, like you can see in these two lines, where eventually you follow the development of the plant, an increasing number of plants become silent until you reach a plateau at 100%. But there were other lines that were showing less triggering of silencing. And what is very important is that, for example, if you take this line that was giving for 45% of silence line, it was the same frequency at each generation. And this was really puzzling, because whatever you harvested the seeds from a silence line or a non-silence, a silence plant or a non-silence plant, at the next generation, it was giving you the same frequency. So this meant that this phenomenon that now we call it PTGS for post-transcriptional gene silencing, but at that time we still were calling it co-suppression. So this phenomenon is not inherited as a silent state, but is inherited as a capacity to trigger silencing at a given frequency at each generation, and this frequency varies from line to line. You have line in which 100% plants become silenced, over at 45, over at 38, over at 20, and so on. Each line has a given frequency, and it's completely determined by the insertion. So not only this phenomenon is dynamic, because also it starts to appear during the development of the plant. It's not appearing suddenly, it's something that progressively increases. So there is, you can never predict when you do a field trial like this, which plant is going to be the first to trigger silencing. You can never predict which one, but this, in the end, the frequency at which it occurs is completely controlled. And not only it's a dynamic phenomena when you look at the population, but it's also a dynamic phenomena when you look within the plant. So by analyzing like these thousands of plants in the field, we could realize that the phenomena was always appearing the same way. It was appearing as a single spot on a single leaf, and you cannot predict which plant, which leaf, which time of the development, but it was always appearing like this. And then the phenomenon was spreading throughout the plant, and it was spreading through the vein first. So the next leaf above this one that was showing this phenomena was just silence around the vein of the plant. And then progressively it was invading more and more the plant until you get a complete silencing at the end. So this phenomena, of course, was, we are thinking that since it started locally and then spread out through the plant, it had to involve a signal that could be not only propagated through the plant, but also amplified. Because without amplification, you cannot imagine that what is happening here could spread in the entire plant, in particular in newly emerging tissues. So to test this hypothesis that a signal was existing, we did very simple experiment. We are using, I didn't mention it, but it was tobacco. At that time, it was the plant model system in the late 80s. And we did this very simple grafting experiment. We grafted a non-silence plant, but transgenic one, onto a silence one, here represented in yellow. We grafted the upper part, which is called the scion, and we grafted it onto the rootstock. And what we saw is that actually there was something moving from the rootstock to the scion that was invading progressively the existing tissue, but more importantly was triggering silencing in the newly emerging tissues. What do we have next? Okay, so we are not the only one who were observing this phenomena. Of course, I mentioned already the petunia and the chalcone synthase, but there are other groups, especially in Europe, who are doing experiments trying to overexpress different enzymes, and who got the same result, and also who confirmed that there was a signal that was existing and that was transmitted from the silence plant to the upper part that was not silenced. So one of the things we wanted to know at that time was whether this was specific to the fact that we were trying to overexpress an existing gene. So what we decided to do is simply to look at what was happening if you introduce something totally exogenous that did not exist within the plant. So we introduced this very simple construct of the glucuronidase, bacterial glucuronidase gene, under the control of the certified source promoter into tobacco first. And we got exactly the same result. We got plants that were expressing the gene, here represented in dark blue, and plants that were silenced. And if we did grafting experiment, grafting a transgenic non-silenced onto a transgenic silence, we got propagation. So this was not specific to the fact that we were overexpressing an existing gene, but simply to the fact that we were expressing maybe under the control of a very strong promoter. I will come back to this later. A very important thing when we talk about signaling is whether this signaling is specific. So we did series of reciprocal grafting, and we discovered that this systemic silencing signal is sequence specific. If you graft a nitrate reductase non-silence onto a nitrate reductase silence, you trigger silencing. If you graft a non-silence Gus onto a non-silence Gus onto a silence Gus, it moves. But if you do reciprocal grafting, nothing happens. So it's really a sequence-specific signal that is moving. Then we were kind of fed up with tobacco. We couldn't do that much. No genetics, long time to get seeds. So we moved to Arabidopsis. Early on, actually almost 30 years ago, we generated these lines with the same construct as the one we introduced in tobacco. And we introduced this in Arabidopsis. And then we got more type of silencing than what we observed already in tobacco. So we observed lines that were non-silenced, represented here as this reference line that we are always using, I will show you later. We obtained lines that were showing post-transcription gene silencing. As you can see, there was still a little bit of activity at the beginning of the life of the plant. You can still detect Gus activity in the cotyledons and in the roots. And we got also lines that were silenced, not post-transcriptionally, but transcriptionally also. So these lines behave totally differently, of course. If you look at simply the expression over the generation, non-silence line, of course, expressed all over the generation. The lines that are silenced post-transcriptionally, just like I showed in tobacco, they lose activity during the development of the plant, and there's resetting during seed formation. And then at each generation, you got silencing that takes place. And it can take place, again, either in 100% of the population or in less. Here I show you a line that shows 100% triggering of silencing, so the activity dropped to 0 during the development of the plant. And then there is resetting during seed formation. And TGS is something that happens more rarely. This transcription silencing, in fact, occurs progressively over the generation, but once it is established, usually after 2 or 3 generations, it lasts and there's never resetting. So there are different hallmarks that are associated to this phenomena. So during post-transcriptional gene silencing, small RNAs are produced, and this is the thing that the group of David Bolcom discovered. You have usually 21 nucleotide small RNA that are associated with post-transcriptional gene silencing. This is exactly what you observe in this kind of line. A majority of 21 nucleotide, a little bit of 22 and no 24. Whereas and you got associated also methylation in the coding region that correspond to the small RNA that are produced. And during TGS, on the opposite, you have no small RNA that are made from the coding sequence because it's not transcribed, in fact. But you get methylation all over the place and actually in the entire locus. So I'm going to mostly concentrate on PTGS. So to decipher this phenomenon of PTGS, we decided, because we were in Arabidopsis, to do a genetic screen. Actually, we did many genetic screens. We did basically, more importantly, 2 genetic screens. One genetic screen, this one first, to try to find mutants that were impaired in PTGS. So we mutagenized this line that shows 100% silencing, and we look for mutants that were expressing the transgene. And on the opposite, we thought that there was maybe a counteracting process existing in the plant. So we mutagenized this line that was not showing silencing spontaneously, and we look for mutants that were showing silencing by post-transcriptional silencing here. So we identified a series of mutants. Mostly, this screen identified mutants that are suppressing PTGS, so they are actually impaired in PTGS component. And here, we identified mutants that are promoting PTGS, so they are impaired in process that counteract PTGS. Mostly what we found here is RNA quality control components. So, to make a long story short, I'm not going to describe the function of all the genes we identified in our screens, but I'm just going to go to the model that trying to explain this phenomena. So how is PTGS triggered in first place? So what we think is that whenever you look at transgenes or endogenous gene, what you got in most cells is transcription of the expected mRNA that's going to be translated into protein. But, of course, you can have also aberrant RNA that are made. So aberrant RNA can correspond to anything that cannot be correctly translated into protein. It can be either something that lack a poly-A tail or lack a cap or is not spliced properly or is not exported at the right place in the cell, whatever. So there is this pathways called RNA quality control, RQC, that is made in the cell to destroy this aberrant RNA, because it could be, for example, if it's misspliced, it could be transformed into, translated into an aberrant protein, which is not good for the cell. So there's a good balance between the amount of aberrant RNA that exists and RQC, and RQC degrade all these aberrant RNA. So in most cells, nothing happened, which is exactly what you see here. Usually in the plant, you don't have any silencing normally. So you could trigger silencing in 2 ways. The first simple way to trigger silencing, and this is what comes from our genetic screen here, is that you can trigger silencing if you knock down RQC. And this makes sense because when RQC is not functional, what you're going to get is no destruction of aberrant RNA. And these aberrant RNA that lack either a cap or poly-A tail, we know that they are good substrate for this enzyme that we identified in our screen that is called RDR6 for RNA-dependent RNA polymerase, which is able to transform a single-stranded RNA into a double-stranded RNA. And RDR6 work with an associated protein, which is an RNA binding protein called SGS3 that comes also from our screen. So if RQC is deficient, aberrant RNA transform into double-stranded RNA. And that's exactly what we observed here when we have mutation in RQC pathway. This line that normally is not silenced, in this background, become able to be silenced and produce small RNA. So the other way, if RQC is not deficient, to trigger silencing is something we have to imagine that happen, is that you're going to get something that saturate RQC. And you can imagine that in one cell, maybe a single cell that we call the initiating cell, you're going to get a burst of aberrant RNA. I'm going to go later on to the hypothesis we have about this burst. So if there is an excess of aberrant RNA of this kind, corresponding, for example, from our transgene, then RQC cannot degrade all these aberrant RNA. So eventually, some of these aberrant RNA that are not degraded by RQC are transformed into double-stranded RNA by RDR6 and HGS3. And then you got an initiation in one cell, maybe, a single cell. So this double-stranded RNA is transformed into siRNA, length of 21 nucleotide in length, or sometimes 22. They can be loaded onto this Argonaute protein, Argonaute 1, that we got in our screen also. And Argonaute 1 can guide the cleavage of the regular mRNA. So if you got only this happening in the initiating cell, it's going to be really localized silencing. But one very important thing is that the 22 nucleotide small RNA that are produced here have the capacity to attract RDR6 and HGS3 to be able to retransform this RNA into double-stranded RNA. And then you get the production of what's called secondary siRNA, really linked to this amplification loop that increase the amount of siRNA within the initiating cell. And one of the very important things is that these small RNA that are made can move from cell to cell and at long distance through the vascular tissues. So something that happened maybe in one cell in the plant can move to the adjacent cell first. And what happens is that this process can be reinitiated. And it can give rise to, again, an amplification loop if you have enough mRNA that is made. But what is very important is that in these cells that are recipient cells, you don't need to saturate RQC anymore. This is only happening in the initiating cell. Once it has been initiated, simply the movement of small RNA from cell to cell can lead to the reinitiation of process, leading to the amplification in the cells surrounding the first one. So this leads to what we see in the first leaf here, which is a diffusion to the adjacent cells. And then what happens later is that since this process is connected to vascular tissue, eventually it leads to the amplification in every cell of the plant. And then you get to this total silencing of the entire plant. So, as Laura said, this kind of movement and spreading of silencing was really corresponding to what happened to when plants recover from virus infection. And this is actually something that a very old paper, as you see, it's from 1928. So we're going to almost very soon we're going to celebrate this the 100th anniversary of this discovery. So this is a very old paper that actually David Bolcombe found in the literature that perfectly illustrates this process. So the recovery process is a process during which a plant that started to be infected at the beginning of the life is progressively recovering from virus infection. The symptoms are, which were very strong at the beginning, are progressively decreasing until to totally disappear. Then you get on the emerging, on the top of the plant, emerging tissues that are totally healthy, totally deprived of viruses. And not only they have eliminated the virus, but on top of this, they are resistant to surinfection by the same virus. If you try to reinfect this plant, now it's totally immune. So this is really, it's type of vaccination, in fact. The plant has been infected first, it has recovered from the virus infection, and then now it's resistant to virus infection. So this process resembles exactly what happened naturally during silencing process. So we wanted to test this hypothesis that PTGS was in fact an antiviral defense mechanism. And immediately, as soon as we got mutants impaired in PTGS, we tested this hypothesis. So it's long ago now, because you see it's from 2000 that we published this. We infected mutants with a virus that doesn't kill the plant, but a virus that is in balance between the resistance and the infection. So if you infect a wild-type plant, you see that you have already a strong symptoms that appear, but the plant is not dead. And if you infect a mutant, then the plant is severely affected and eventually it dies. This picture was taken before the plant died. And the plant died because it's no longer able to control the amount of viruses. As you can see here, this is the RNA from the virus. Here in this case it's CMV. Sorry. You got a certain amount of virus that is present in the wild-type plant, but in the mutants, either here RDR6, SGS3, or the double mutant DCL24, then you get a huge amount of virus that accumulate that eventually kill the plant. So PTGS is really a mechanism that is active against the viruses. So what about endogenous gene now? I showed you a model in which an aberrant RNA can be transformed into double-stranded RNA and then into siRNA, and then this siRNA can spread throughout the plant. Of course, in the model that I showed you, endogenous genes are also able to produce aberrant RNA. So the question is, why does PTGS is not affecting endogenous gene also? So the first question we ask is whether endogenous genes are able to produce siRNA. So in wild-type condition, here in the wild-type Columbia accession of Arabidopsis, if you look at protein-coding genes, only protein-coding genes, you don't find any small RNA of 21 or 22 nucleotide length, long in length. Here, the red dot, there's nothing at all. But when you mutate RQC, and here we mutate two different components of RQC, VCS here and DCP2, then you start to see a huge amount of 21 and also a little bit of 22 nucleotide small RNA that are appearing. It's a huge amount. And these come from thousands of protein coding genes, thousands of them. And we know that most of them are really siRNA coming from PTGS because they are RDR6 dependent. Here's a metaplot distribution of small RNA coming from mostly from the 3' of the gene. And if you are looking at double mutant between the RQC and RDR6, you lose this small RNA. So they are actually coming from both strands. And very importantly, RQC is, we know that it's really essential because if you mutate RQC, most of the plant die. As you can see here, VCS mutants or DCP2, they just germinate and immediately die. They don't go further. And you can rescue this mutant, partially only, by mutating PTGS behind. Here you can see at the beginning of the plant is really making true leaves, and eventually it grows. It's still impaired, but it grows. So it means that RQC is essential to destroy aberrant RNA, because if aberrant RNA are not destroyed, they are transformed into double-stranded RNA by PTGS, and this leads to the death of the plant. So, but why is PTGS not spreading out throughout the plant when you target endogenous RNA? This is very important because you can imagine easily that RQC can dysfunction in certain cells once in the life of the plant. The probability that you have a dysfunction of RQC in one cell of the plant during its entire life is very high. So what could happen if RQC was dysfunctioning in one cell and then you got spreading? You would kill the plant each time there is a dysfunction in one cell. So of course this should not happen. And actually we know that endogenous gene cannot undergo systemic PTGS. We know it from various experiments. We know it from experiments we've done with tobacco. When we were silencing the endogenous nitrate reductase using a transgenic nitrate reductase, we knew that silencing was propagating. But when we were grafting just a wild-type plant that contained the target, the endogenous nitrate reductase, when we were grafting this, nothing was happening. So this means that endogenous gene cannot amplify PTGS. And we did this for another gene, another endogenous gene called NAIA, encoding for another enzyme, and the same thing happened. And also the group of David Bolcombe and the group of Olivier Voinet did experiment where we were inducing silencing locally in the companion cells of the phloem using an inverted repeat. And what they showed is that silencing was just moving to the adjacent cells, but never further away, meaning that there was no amplification at all of endogenous gene. So why is that? It's very simple. It's a question of level of aberrant, or of not aberrant RNA, but the level of target RNA, the regular mRNA that is targeted by the small RNA. There is, in fact, an amplification threshold that exists. If you are above this amplification threshold, if you have a large amount of mRNA that is targeted by the initial small RNA, not everything is destroyed. A part of this target mRNA can be reused to produce secondary siRNA, and then the amplification loop occurs, not only in the initiating cell, but in the adjacent cell, all the recipient cells of the cell or of the plant. So if you are expressing above this amplification threshold, then you got systemic spreading. And in fact, this can only happen when you got a huge amount of target mRNA. And this huge amount of target mRNA, you can only get it when you use a viral promoter, because viruses are amplifying so much in the plant that when you use this viral promoter, you can get a huge amount of mRNA. But when you target endogenous gene or transgene expressed under endogenous promoter, you are below this threshold level. We quantify the amount of mRNA that is made by a viral-driven promoter compared to an endogenous gene. It's above every endogenous gene. So there is really this threshold. And what happened when you, this is what happened with transgene. This is what happened, in fact, when you target simply an endogenous gene. All the siRNA that are made here in the first initiating cell are moving here, but there is no amplification loop occurring because there is not enough mRNA. All the mRNA that you have here are used and destroyed, and there is no secondary siRNA that is made. So all these siRNA that are moving from the initiating cell to the recipient cell are simply consumed for targeting this. And this doesn't go further away. So this RNA consumption results in a limited movement of small RNA from the initiating cell to the adjacent cell, and then it stops. It doesn't go further away. And this is a chance for the plant that not any endogenous RNA is making enough, any gene is making enough mRNA to get to this amplification loop. Because, again, I said, if RQC was deficient in a single cell, it would spread out everywhere. And this also explains why there is a very limited number of examples of natural PTGS cases existing in nature. And each time, they affect non-essential genes, because of course if it was affecting an essential gene, the plant would die immediately. You have these very few examples of gene duplication, actually natural duplication of chalcone synthase here in petunia or in soybean. These are not transgenic plants. They are simply natural variety existing that either had undergone duplication here, leading to this phenomenon of silencing, or in the opposite case, you have a variety of soybean that is colorless seeds, because there are clusters of 6 CHS genes which lead to silencing of the gene, so there is no CHS produced in the seed coat, and then a deletion can lead to the reduction of the number of chalcone synthase. There is no more double-stranded RNA made, and there is color in the seed. So now, how many time do I have still? 15. Okay, good. Now let's go back to this first question mark. How can this happen spontaneously in a transgenic line? How can silencing be triggered spontaneously? Of course, I told you it can happen when RQC is deficient in one cell. But if it were the case, every transgenic line should undergo silencing at the same frequency, because the chance that RQC is impaired in one cell is the same for one plant or another. So the fact that every line has its own frequency of triggering means that something is special to each locus. So there must be something happening at each locus with a certain probability. So what is happening at each locus that gives rise to a burst of aberrant RNA at a different frequency? This is something that we wanted to find. And first thing we try to look at is where is actually silencing starting. So this is very old pictures now. With this very old Northern blot, we realized that silencing was always started from the 3' end of the transgene. Sorry, and this is also very easy to visualize when you do RNA-seq. There is always small RNA starting from the 3' and then move progressively to the 5'. So we said, okay, maybe it has to do with the fact that we have something happening on the 3' of our transgene. So we made a lot of construct. At the beginning, we were working with this construct here that has a selectable marker oriented in the opposite orientation to the 35S Gus gene that we are following. So we said, okay, maybe there is simply transcription that is going to read through transcription that is going here, that is making double-stranded RNA because you have sense and antisense RNA that are made. So we changed a lot of things. We did a lot of construct. We changed the orientation of the selectable marker here just by flipping this region. We changed the selectable marker by itself, the promoter driving the selectable marker. We even made transgenic plant without any selectable marker that we just screened by PCR. We changed the terminator because the small RNA are started in this region. We changed the terminator. We change the terminator without any selectable marker, and we use even another endogenous terminator. And in the end, didn't change anything. Silencing was occurring in the same way, whatever construct you were using. So it has nothing to do with the type of construct that you're making. Okay, so again, the genetic screen told us a lot of things. In our screen, I told you that we got mostly PTGS component or RQC component that prevent PTGS. But we found another category of mutants. And these mutants are DNA repair mutants. And in fact, when mutagenizing this line that never shows silencing spontaneously, we found that many mutations in DNA repair factors are promoting PTGS. Actually, they are able to initiate PTGS where normally there is no PTGS occurring. So this means that impairing DNA repair could promote PTGS. So we thought, what does it has to do between DNA repair and gene silencing? We thought that maybe DNA damages could trigger actually PTGS. So fortunately, there was this wonderful tool that appeared, which is CRISPR-Cas9, where you can determine exactly where you want to cut the DNA. So what we did is simply to test the hypothesis that DNA damages was actually the trigger of PTGS. So we introduced double-strand break downstream the Gus reporter, because of course we do not want to mutate a Gus. Of course we would get not silencing but mutation. So we use guide that cut the DNA behind the poly-A tail, so we would be sure that there would be no impairment actually of the transgene. And what we did is that what we observed is that each time we cut the DNA, we got silencing. Here's the control of a line transformed just with Cas9 and a random guide. And this is what we observed with these two guides. Almost 100% of the plants are silenced. So is it really PTGS? Yes, it is PTGS because in these plants we can detect small RNA, exactly like what we detect, for example, when we mutate RQC. And the confirmation that it is PTGS is the fact that this phenomenon does not occur in an RDR6 mutant background. When you use this guide, it clearly shows that this has no effect on the transgene. You got no silence plants in an RDR6 mutant background and no small RNA that are made. So it means that when you cut the DNA, what you obtain exactly in the vicinity of the cut is the production of small RNA. Okay, we ask whether this phenomena can occur at a distance from the transgene. So we tested a guide that were cutting the DNA either very close to the Gus reporter or further away. Each time you are very close to the Gus reporter, 1, 2, and 3 here, this guide, you get 100% silencing. And as you get further away, the efficiency of silencing decreases until something very low, but still existing, even at 2 kb from the gas reporter. Is it really okay? Is it really a double strand break that is necessary? We asked this question by testing derivative from Cas9. You know that if you mutate Cas9 in a way that you transform it into a nicase, you get, instead of a double strand break, you get a single strand break. And if you use dead Cas9 that just locate on the DNA but doesn't cut, yeah, there's no cut. So we use the same, we associate these different Cas9 derivative to the same guide. And as you can see, only when you make double strand break, you get silencing. When you use the nicase or the dead Cas9, you got no silencing. So actually, it's a double strand break that initiate PTGS. So what do we think about, how can we explain this? How can we reconcile this CRISPR-Cas9 experiment with what is happening naturally? What we think is that the frequency at which a given transgenic line undergoes PTGS at each generation depends simply on the probability that a double strand break occurs in the vicinity of the transgene in at least one cell of the plant. So, of course, every cell can undergo DNA damage. The cells of the plant are exposed to various things that can trigger DNA damage. So, if a double strand break occurs in the vicinity of the transgene, it can trigger silencing because once it has been initiated, it's propagating through the entire plant. What we think is that when a double strand break is made, there are aberrant RNA that are produced. And actually, we just got RNA-seq result very recently that shows that at the DNA break, there is actually aberrant RNA that are made here. And of course, these aberrant RNA are probably the ones that saturate RQC, leading to PTGS. And the reason why the frequency is different from one line to another is simply that the probability that a double strand break occurs in the vicinity of the transgene is different from one locus to another. It's simply as simple as this. And here it means that in this type of line, you've got 100% chance that at least one double strand break occurs in one cell during the life of the plant. But here you have loci that are more resistant to double strand break, and there is a probability that this happens is much reduced compared to this line. So, another question, of course, is what happened to endogenous gene? Of course, endogenous genes are also exposed to double strand break. Or actually, are they also exposed to a double strand break? It's a question mark. So I told you that already endogenous genes are protected against PTGS by the fact that they are not producing enough mRNA to lead to the amplification loop. So to know if double strand break can initiate PTGS at endogenous gene, we have to use a trick to force endogenous gene to undergo amplification. And for this, we have this trick is to use a DCL4 mutant, because when you mutate DCL4, you stop the production of 21 nucleotide siRNA. You force the production of only 22 nucleotide siRNA through DCL2. And then you force the amplification loop to occur. So what we did is that we targeted our favorite endogenous gene, which is nitrate reductase. So we introduced double strand break downstream the nitrate reductase gene in Arabidopsis. This is a wild type Arabidopsis, Colombia. This is wild type Arabidopsis with CRISPR downstream. There is nothing happening. But if you work in a DCL4 mutant background, you see that DCL4 mutant grow perfectly well. But if you target NAE with a double strand break with Cas9, then you get 100% silencing happening, meaning that actually endogenous gene can trigger PTGS when they are targeted by double strand break. Another gene that we used, BAC1, which is another gene for which the silencing is quite evident to observe. This is a wild type plant. This is a BAC1 mutant. if you introduce double-strand break downstream back one in a DCL4 mutant background, then you can recapitulate the mutant phenotype. So it means that each time you introduce double-strand break, you can trigger PTGS as an endogenous gene. Okay, I don't think I have time to go through this. Unfortunately, I wanted to show you just briefly that we have time? Okay, good. Good. Excellent. So, PTGS is always triggered when we target transgene driven by the 35S promoter. So we wanted to know what happened if you use a transgene that's driven by an endogenous promoter, such as the ubiquitin 10 promoter, which is kind of a strong promoter, but not as strong as the 35S promoter. So we use the same strategy of introducing double-strand break downstream. And what we observe is that, we got silencing again, using guide that cut here or even downstream. So each time we look at GUS expression, and you see almost all the line, more or less efficiently, are silenced. We said, okay, this works for almost everything. It works for 35S driven transgene, it works for endogenous gene, it works also for a GUS transgene driven by an endogenous promoter. But of course, we did all the controls. RDR6, it works in RDR6 also. We said, oh, what is this? We have a new kind of silencing that's happening here because it's not RDR6 dependent. So what we did also is to look at the inheritance of this process. You remember I told you that PTGS is not inherited per se. What is inherited is the probability that it's triggered, whereas TGS is inherited. So when we look at the inheritance of this phenomenon, we segregate the Cas9 away. If it's PTGS, the line that lags the Cas9 will simply express the transgene. But if it's TGS, it's going to be maintained. So in this case, when we get silencing in a T1 here, like this, if we look at the progeny, the plant that lack the Cas9 are still silenced. And in fact, it's getting not only their silence, but we even found lines in which we have a totally wild-type sequence. So this means that it's clearly not mutation that is impairing the expression of the transgene, because here, the sequence has been cut and repaired to the identical. It's simply the fact that it has been cut and it has imposed a new epigenetic state that is actually inherited. And when we look at we are now at the T8, you see that progressively, that's exactly what you observe when you get TGS normally. So we get another type of silencing that can be triggered upon DNA damage triggered by CRISPR. So it resembles exactly what we observe naturally when we have a line that undergo TGS. It's progressively silenced over the generation. So of course the hallmark of this is pure total methylation of the transgene. We looked here at what happened in our locus. Here we have a BASTA-resistant reporter. We see that this line that has been silenced also is almost completely silenced for the bar gene. And if we look at methylation, the level of methylation is 0 in the original line. If you look, whatever you look at the promoter or the GUS coding sequence, here you have no methylation at all in the control. But when you look at the line that we obtained after CRISPR cut, we got full methylation almost in the promoter and even in the coding region. So this explains that it could be TGS, and actually it is TGS, we know it, because we can reactivate the transgene by the classical way to alleviate TGS, either by treating plant with 5-acetidine or treating it with the DDM1 mutant background. We can always reactivate the transgene. So, is it specific to this locus? Again, we wanted to make sure. We tried 2 other loci with different type of transgene, different arrangement. Each time we cut, we got silencing also appearing in the ER6 mutant background. So, I'm just finished now. So what I can tell you is that, beside the fact that we have made a new hypothesis to explain how PTGS is naturally triggered in transgenic line, I would like to draw your attention that you must be very cautious when you do CRISPR experiment. Because so far people have thought about CRISPR as a wonderful tool, and it is a wonderful tool, for sure. People are concerned about the side effect and the off-target effect of CRISPR, thinking that there might be mutation appearing somewhere else in the genome, beside the target you want to modify. But there might be other epigenetic changes that occurs. So of course, if we want to make a knockout, it's not a problem, because whatever you make a mutation, and on top of this you make a silencing, it's no problem. It's even much better. But when you want to do changes, site-directed mutagenesis or gene replacement or whatever, you're going to get things that you can't explain. Because in fact, if you replace one sequence by another, and then this new sequence is epigenetically modified, you don't know if the phenotype you observe is due to the mutation you have introduced or to the epigenetic change that occurred. So it's very difficult. And I have a colleague working on moss who did this experiment, because of course in higher plants it's very difficult to do gene replacement. So in moss, what he did is that he replaced an endogenous gene by a GFP reporter. It's easy because in moss you have homologous recombination, so it works 100%. And every GFP replacement that he got, where replaced the endogenous gene by the GFP, is identical at the sequence level, but each of them express GFP at a different level. So it means that the day we are going to be able to do gene replacement or site-directed mutagenesis using CRISPR in higher plants, it's going to be difficult to figure out what is the cause of the change you observe. And of course, there might be different type of change. All we know now from our experiment is that when you get a transgene driven by a viral promoter, what happens mostly is PTGS. So far, what we've done when targeting endogenous gene is also PTGS. But we haven't tested a lot, of course. We have tested, I show you 2 examples, but we're in the process of testing more now. And surprisingly, when we got an endogenous promoter driving us, we've got TGS. So this is, we are just at the beginning of trying to understand what's happening here. We don't know why, in some case, we got TGS and why we got PTGS. The only thing we can say is that we have to be very cautious when analyzing CRISPR event, because not only we have to have concern on the gene that we are targeting, but we should have concern also on the genes that are surrounding this target. Because as I showed you, 2 kb, when you do a double-stranded break 2 kb downstream your gene of interest, you have a modification. So this means that reciprocally, when you target this gene, the other next one, can be also affected. So we have to be very cautious in this case. So with this, I'm going to end. I want to show you my group, my current group. Especially I want to thank Tallinn Elmayon, who has been working with me since the very beginning of this project. It's been now 32 years ago that we've been working together, and in a few years we're going to retire together also at the same time. And other people, and especially also for all the CRISPR experiment, a very talented PhD student, Martin Lacroix, who did all the experiment I presented in the last third of my presentation. So with this I end, and thank you for your attention. I would be happy to answer questions. Thank you.
Thank you very much, Hervé. And now it's time for you. Who is the first hand who requests? Okay.
Impressive talk. Thank you. I'm wondering about the concern you raised at the end of your talk about when we do CRISPR, mostly for changing, not for mutation. If I understood well, you said that this post-transcriptional gene silencing or the transcriptional gene silencing occurs most probably when the gene has a higher expression, right? So is there a threshold of, I mean, if I'm targeting a gene below a certain level of PKM, I'm safe? Do you?
I think so, yeah. I think when you post-transcriptional gene silencing is not a concern when you're targeting endogenous genes.
Okay.
Because every endogenous, every endogenous gene is below the threshold that can lead to PTGS amplification. So I think PTGS is not a concern really, not only because the mRNA level is too low, but also by the fact that it's not inherited. If you segregate Cas9, the concern with PTGS is not really a strong concern, I would say, except if you look only at T1. If you look at T1, you can have things that affect the surrounding genes, or even maybe genes that are interacting through 3D interaction. I mean, the major concern is TGS, because it's something that is inherited after elimination of Cas9. So PTGS is just a caution that you should have during the T1 observation. When you, for example, you look, you've got a series of plants, you say, oh, this one is the most interesting. Be careful that you go until the T2. But for TGS, it's much more a concern, because it can be really inherited, and actually it can still increase after elimination of Cas9, which is something we do not understand what's the trigger. Actually, for PTGS, we have a kind of good understanding now of how it's triggered, but TGS is still a mystery. How can something be triggered progressively during generation? It means there are some kind of layers of mark that are put, epigenetic mark that are put on the DNA and the chromatin, and passes through meiosis, and then until you kind of saturate the system.
Yeah, amazing talk. Really well explained. I wish some of my students were here, not just, you know, PhD, but also at the school, to understand all this. So the main concern when you make a CRISPR is the off-target. So you make a bunch of, look at the genes, homologous, whatever. But then this means that you also have to take care of genomic regions that is close in the 3' end of the gene, and then kind of, because then you can start making, you know, you will never find that out. So you can start making, breaking in 3' end of a gene that will be silenced in this.
It seems that one thing I didn't mention is that the major concern is whether you could produce antisense RNA, in fact. I didn't show it, but when you target, for example, upstream from the transgene, you don't get silencing. So really, it's in the model. If I go back to the PTGS model, yes, this is it. Oh, too far. Yeah. When we break the DNA here, for example, we don't have silencing. So it's really, when you get double-strand break, you have concern on the gene here only if it's oriented in this way. Yeah, I think so. I mean, of course, we cannot, like always, we cannot exclude anything. In biology, everything is possible. So, but you can also imagine that you have R loop or things that make a close contact with things, and that transcription could actually occur kilobase away from this. So it's possible.
I can only imagine with all these lines of tDNA lines we have been using, you know, forever, that they can be actually really messy, right? Because I know they are. I want to talk to you about this later. If you know, I want to talk to him. So.
Aborted insertion that simply depose some epigenetic marks, like we saw from this line, for example. For us, the most striking is this. We have a wild-type sequence, and we had a complete loss of expression. So this means that the DNA has been cut, repaired. So when you get tDNA insertion, it's the same thing. You have plenty of aborted insertion that are just repaired, almost even to the identical. So there might be plenty of things that happen during tDNA insertion that either cause mutation due to the insertion, or due to the effect of the insertion to the surrounding genes, or even to epigenetic changes occurring during DNA repair, simply. Yeah, tDNA insertion lines are just a mess. I never use them. Except for studying silencing.
Thank you very much for a wonderful talk. So My question is, in your model, you distinguish between viral promoters, endogenous promoter, but if you use an endogenous promoter, I mean a promoter from endogenous gene that has really high levels of expression, I don't know if you can reach the levels of a virus, not probably.
You can get some lines that are close to the level of the 35S promoter. And actually, we've been looking for lines like this that spontaneously trigger PTGS. And what we found is really strange. It's also something that we are still trying to figure out what's happening. We know that these lines are initiating PTGS, probably because there are natural DNA damage that occur, but they never amplify PTGS. And probably because the ubiquitin promoter is just below the level. And actually this threshold, you know, it's really, can I go to this? This threshold level is really tight, you know. It's really something really, really precise. And it seems that none of the endogenous genes can reach this threshold. Because, for example, we have a line that is just expressing here, transgenic line, 35S here. In a homozygous state, it's expressing here. In a hemizygous state, it's just below, no PTGS at all. So it's really something really, it's just like if nature has really selected a balance between the efficiency of RQC to prevent aberrant RNA to be transformed into double-stranded RNA, and a precise threshold level of all the endogenous genes so that they never get amplification due to the action of DCL4. Because if you remove DCL4, you can naturally get silencing occurring on endogenous gene. So it's a perfect balance between RQC and the ratio between DCL2 and DCL4.
I have a question related to that. If that is true, and I have no doubt that it is, then when an organism, I mean when a plant in this case, needs a lot of protein of a protein product of a specific gene, there is, let's say, a natural limit in the strategy that the plant can use because it cannot be through heavy overexpression at the transcription level. It has to be by very strong translation, because if you do too much transcription, then you trigger this. But then other organisms don't have this amplification system, so that would actually shape the way in which the different organisms make their strategy to overexpress a protein when they need to.
I've never thought really about this question. So, but I get clearly, yeah, every organism has found its strategy to fight against. It's very clear that double-stranded RNA has not the same outcome in organisms that don't have an RDR, for example. So it's very clear that something is happening here. Yeah, I can't really answer your question simply. Yeah, it's kind of evolutionary things. Yeah, but already in plants, among the different type of plants, there are really different behavior. I mean, the balance between all these phenomena, TGS, PTGS, the importance of the different components is clearly different between monocot, dicot. It seems to be kind of a general trend, but of course, with every plant, every organism is unique and has its own evolution. It's very funny. But it's people always ask me, what about endogenous genes that are expressed at very high level, like Rubisco, for example? If you look at Rubisco, it's very amazing because first, Rubisco is below the threshold. If you compare the 35S Gus line that trigger, it's above Rubisco. So there is nothing that, even Rubisco or LHP, I don't remember the name, all these photosynthesis genes that are highly expressed, none of them reach the level of the 35S. So it's very clear that there is this. The other thing that is amazing is that when you look at, where are the endogenous genes? Yes, when you look at the endogenous genes that produce small RNA, the genes that are you could think, okay, the more the endogenous gene is expressed, the more it's going to be able to produce siRNA. No, no, no. Rubisco is not part of it. So it means that there's another layer of control. The genes that seems to be the most highly expressed are the less prone to produce siRNA, meaning that probably they are away from double-strand break. They are in regions that are resistant. And that's actually something that we are trying to investigate. We're in the process of sequencing about 100 transgenic lines that we have characterized that trigger PTGS at different efficiency, even the ones that are strongly silenced and the ones that are kind of resistant to silencing, let's say. It happens so far that the lines that are triggering the most silencing are always in intergenic region. And the lines that trigger the silencing the less are within genes. Just like the probability to get double-strand break within genes is less than in intergenic region, which makes sense also. So this would explain why I said that thousands of endogenous genes are producing siRNA in RQC deficient mutant background. Not all of them. And it's amazing to find that Rubisco and all these strongly expressed genes do not. And of course, it makes sense also that during evolution that everything that could be deleterious for the plant would be lost. Simply.
Okay, thank you very much again for your nice talk and also so many questions and answers. Do you want to make another question? No? No? Okay, let's give him another applause. Thank you very much. Enjoy Andalucía.
I wanna dance by the water beneath the Mexican sky. Drink some margaritas by a string of blue lights. Listen to the mariachi play at midnight.